Hello, everyone, and welcome to today's webinar, Medical Laboratory Performance Benchmarking, Current Practice and Future Perspectives. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Abbott. To learn more, visit corelaboratory.abbott. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Dr. Wolfgang Huf, Medical Director, Vienna Healthcare Group, Klinik Heizing, Vienna, Austria. Dr. Huf, you may now begin your presentation.
Thank you, Marie, for your kind introduction, and welcome again, everyone. I've roughly structured this talk into general background, discussion of a recent study and perspectives for individual laboratories. First of all, my personal story. Why am I such a fan for benchmarking? The short answer is it helps to make good decisions. The long answer begins with me being a general practitioner and regarding laboratory standards as very high, in particular in the age of evidence-based medicine. Then I received training as a laboratory professional, which subjectively confirmed my view regarding standardization in laboratories. But I also found that there is much room for development. In particular, there is a lack of peer-reviewed data for management decisions. What is benchmarking? How can we define it? A benchmark is something that serves as a standard by which others may be measured or judged, according to Merriam-Webster. Quality is a relative concept. As it is said in medicine, today's state of the art may be tomorrow's grounds for a trial in a court of law. To assess whether we deliver good quality, we need a reference. In clinical medicine, we use guidelines. In the laboratory, we don't have much of that, in particular regarding management decisions. This is where benchmarking comes into play. The laboratory traditionally focuses on measurement quality. There is a host of external quality assessments available and widely established. However, there is also an increasing need to improve quality in the interaction with the hospital and management decisions, in particular regarding financial sustainability. In PubMed, the standard database to research on this, there is little to be found. When you type in medical laboratory and benchmarking as a search term, there are 34 hits to be found. You can find more on the term EQA, but this is primarily relating to topics of measurement. Right now, a lot of international conferences focus on the so-called pre-analytical phase. This is the phase where about 65% of laboratory errors occur, and about 65% of costs incurred by the laboratory are generated. So there is a need for data relevant to management decisions. But why isn't it here already? As I said, benchmarking hitherto mostly focused on instrument and assay performance. There are a few international studies trying to compare different laboratories in different countries, different regions of the world, and they most rely on so-called LIS data, Laboratory Information System data. This is in particular the case for the IFCC, the International Federation for Clinical Chemistry, working groups on laboratory errors in patient safety, which extracts most of its data from data that is generally to be found in the laboratory information system. There are some questions that need to be extracted by hand from different sources. And there are a few studies that focused on management approaches, but they have not yet published questionnaires. Indeed, we didn't find any published questionnaire regarding worldwide approaches to quality in laboratory management. So there is a need to generate and publish a questionnaire. What do we need to do in particular? Evidence-informed decision-making is rapidly becoming the norm in medicine. There is the so-called European Union HOPE program. HOPE is the European Hospital and Healthcare Federation. 2019, the theme was evidence-informed decision-making in healthcare management. And this year's theme is using evidence in healthcare management. So it directly relates to the 2019 theme. The basic idea is to use reliable data to support decision-making in management. You cannot identify the exactly best decision, of course, but the more data you have, the better informed your decisions in general are. So we need to recognize the status quo in comparison to others. And the academic approach to do this would be to first design a questionnaire, then design a reliable data elicitation process, validate both in the pilot study, and then use them widely. In particular, we should use the PDCA cycle, the Deming cycle, plan, do, check, act, to continuously improve such a questionnaire. This idea was the basis for the medical laboratory benchmarking that we published recently. Now, why do we need a questionnaire posing quasi-redundant questions to elicit reliable information in the end? Directly estimating one's own performance in relation to others is not the strength of the human mind. As you can see in the study we conducted recently, about 98% of laboratories rated their performance at or above the industry average. This is mathematically impossible for normally distributed data. Now, having sketched the general background, I will proceed to talk about a recent study and narrow down on the challenges and opportunities associated with benchmarking. A benchmarking study relevant to laboratory management needs to cover three key aspects: the process dimension, the clinical dimension, and the financial dimension. I will now present a few questions that were deemed relevant by a group of experts. And mind that this questionnaire has already been published. You can see the citation at the bottom of the slide and can always look it up. In the process dimension, three examples for questions for items actually used was, does your laboratory measure defined key performance indicators? Examples given were employee productivity, workspace utilization, the amount of implemented auto-validation rules, and so on. Another example is, which of the following turnaround times displayed below does your laboratory monitor? Turnaround times can be defined in various manners. Most of the laboratories use as their turnaround time the so-called lab turnaround time, which is defined from the beginning of sample reception to the end of result release. The most interesting turnaround time, of course, is the so-called total turnaround time that starts with the order and ends with the result release. A more comprehensive approach still is the brain-to-brain turnaround time that starts with order and then ends with physician's access to the data. The third example was, do you use any of advanced IT functionalities listed here? These were, for example, smartphone-related alerts and applications, instrument capacity monitoring, and information dashboards to display KPIs. For the clinical dimension of laboratory benchmarking, example questions are, which of the following services does your laboratory provide to physicians? For example, results and interpretations in combination, proactive consultation on complex patient cases, or diagnostic pathways guidance. How regular did your laboratory and clinical care teams review the diagnostic pathways or guidance together in the last 12 months? Or is your laboratory combining data digitally with other diagnostic disciplines? A very interesting item was also the question regarding the laboratory measuring impact on patient outcomes. This is particularly seldomly used. The last but not least dimension is the financial dimension. Most of the laboratories do not like to give away the direct financial results, but for a benchmarking study to be relevant, we need to have some estimate about the financial dimension. And items deemed relevant by experts were what percentage of results are auto-validated, how many full-time equivalents were used to operate the analytical workstations, or the physical space needed to operate the analytical workstations. As our goal in designing a study of this kind is generating data that fulfill the so-called measurement quality criteria of objectivity, reliability, and validity, we trained a host of professionals to identify valid data in direct one-on-one conversations. An online questionnaire was filled out by heads of laboratories and these trained professionals together, which assured confidentiality and anonymity. Of course, there was a possibility to give contact data to get individual feedback. The design process of the questionnaire was iterative. First of all, there was an extensive literature research, and then we used author experience and the input of informal focus groups of laboratory professionals to reduce the number of items in the questionnaire to 50, in order that the questionnaire was short and could generate high participation and high completion rates. As mentioned, it was essential to train professionals to assure consistency and confidentiality with pseudonymized data. The resulting questionnaire consisted of the 3 dimensions: operational performance, integrated clinical care performance, and financial sustainability. There were some items regarding general information at the beginning. Altogether, 50 items. 773 laboratories answered the survey. As far as we know, this is the largest survey to date regarding laboratory performance in management. The top 10 participating countries were Italy, Russia, Saudi Arabia, Spain, United Kingdom, Germany, Greece, United Arab Emirates, South Africa, and Serbia. Altogether, about 60% of laboratories were government hospital laboratories, about 1/5 were private commercial laboratories, and about another 1/5 private hospital laboratories. There were a few research laboratories, testing laboratories that were excluded in the further analysis. Now, how do laboratories rate in the 3 dimensions? In all 3 dimensions, laboratories were about normally distributed, which is a prerequisite for the statistical analysis we performed afterwards. This is so-called factor analysis, which more or less automatically sorts the individual items used in the questionnaire into different subdimensions. In the subdimension of operational performance, we found 7 items. In the subdimension of integrated clinical care performance, 6 items, and in the subdimension of financial sustainability, we also found 7 items. You can identify them here. And this is basically the central figure of the study, the central plot. You can see that there is a correlation between operational performance and integrated clinical care performance. Financial sustainability is coded in green, amber, and red. There is a group of laggards in the lower left-hand quadrant and a group of leaders in the top right-hand quadrant. But the interesting thing is the deviation from this rule of correlation, which might be of interest for individual laboratories. There is, for example, one laboratory which is indicated with the blue arrow that is in the upper right quadrant and rates very high in operational performance and rates very high on integrated clinical care performance, but is in the lower quartile of financial sustainability. For the individual laboratory, it might be of interest to identify the things that could be optimized to increase financial sustainability. And this can be done in a sub-analysis. In this case, the laboratory, among others, had practically no auto-validation in all the laboratory specialties like clinical chemistry, immunoassays, hematology, urinalysis, coagulation, bacteriology, routine molecular, and specialized molecular. In this second example, we examine a laboratory that is above the median in this case of integrated clinical care performance and financial sustainability, but is a laggard in a subdimension of operational performance. Again, we can examine the reasons for this, and we would find that the laboratory doesn't measure patient satisfaction, doesn't measure clinician satisfaction, and doesn't measure employee satisfaction. Furthermore, it doesn't have any procedures relating to Lean or Six Sigma. The third example is a laboratory that rates relatively high on operational performance and financial sustainability, but rates rather low on integrated clinical care performance. The reason for this is, for example, that it doesn't combine any data digitally with other disciplines. So what can be done in general for an individual laboratory? I have now discussed the background of benchmarking. I have discussed this study, but for the individual laboratory, we need some action items. The typical approach for your lab would be to identify the subscale or subscales of interest after participating in such a benchmarking study, identify the items of interest in the subscales of interest and its relative position to other laboratories in the country, for example, or in the specific laboratory community like hospital laboratories or commercial laboratories, and then discuss with staff and make plans for the coming year how to improve on these measures. Ideally, a PDCA cycle should be started. The concrete action trajectories are regarding the allocation of existing resources or the generation of new resources, budget requests, etc. The general benefits of the benchmarking approach are that benchmarking allows quantification and thus comparison, and ideally business case generation. Always remember that top management needs data to do investment and allocation of resources, and they even might need some arguments for shareholders. This is the publication. To sum up, the background was that there is a historical lack of global initiatives on collecting and benchmarking management data for diagnostic medical laboratories. We did a study that was published in the pilot you saw in the slides above, and found its follow-up study in the study published here in Clinical Chemistry and Laboratory Medicine that gathered data from all around Europe, Middle East, and Africa. Its aims were to establish a good practice approach towards benchmarking, to generate a snapshot of the current state of the art in this region, and most importantly, to set the stage for global follow-up studies. Your immediate reaction might be how to participate in these global follow-up studies. You can participate using this link here. And for more background information, visit the core laboratory site at Abbott. This was it from my part, and I'm ready to take questions now.
Thank you, Dr. Huf, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located at the far left of your screen. We'll answer as many of your questions as we have time for. So let's get started. Our first question is, how can I be sure that my data are kept confidential?
Well, per default, data acquisition and processing in this study are of course anonymized. And there are several regulations like the General Data Protection Regulation of the European Union and other relevant regulations that are naturally adhered to. But of course, there is the opportunity to get individual feedback if possible and decide by the participant. In this case, contact details can be given and data will be returned to the participant, but data analysis per se is always done with anonymized data.
All right, thank you. Next question. Since data are anonymous, can I be sure that they are representative for the laboratory landscape?
First of all, representativeness is a relative concept. It depends on the subgroup you're examining. And of course, there is some potential for bias in any study. Nevertheless, I already mentioned that the study presented here is the largest study to date. So it is probably the best you can do right now. And regarding utility, which is the most important question for each and every individual laboratory, it's like with the telephone. The more people you use it, the more benefit you might have. And I hope that this webinar will also increase participation and thus the utility for you.
Great, thank you. Next question. How exactly do you envision benchmarking to continue in the future?
I believe benchmarking will be much more central in the future of laboratory medicine. In particular, I think that each laboratory will probably not only have a sort of chief benchmarking officer, but also a high degree of benchmarking literacy in the entirety of its staff. To this regard, we are currently running the follow-up study, the links to which you can see on the screen, and promoting knowledge about the utility of benchmarking on an international scale.
All right. And it looks like we have time for one more question. How has the benchmarking data helped your lab?
Well, the way benchmarking data helps the individual lab depends to a high degree on the current state of development of this lab. You saw in the scatter plots that individual laboratories have quite a varying degree of, so to say, maturity. In our particular lab, we felt the need to increase automation and also focus more intensely on the pre-analytical phase in concordance with international conferences and their focus on the pre-analytical phase. Of course, this is known by most professionals. You most of the time know the needs of a laboratory, but you don't have the data to prove it in discussions with the management. So the benchmarking study was a wake-up call for us all. And we now strive to achieve a fully digital ordering process. As a side note, during the current pandemic, the city of Vienna has impressively demonstrated a full electronic ordering process for the masses. So it should be achievable by any laboratory worldwide.
All right, thank you, Dr. Huf. Do you have any final comments for our audience?
Well, I enjoyed the webinar and thank you for the great questions. Have a nice day.
Thanks. Thank you again, Dr. Huf, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Abbott, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.
Goodbye.